Good evening, everyone. I'll start the session in another 2 to 3 minutes. Hello, and I welcome you all to this week 8 session for problem solving of the NPTEL course Cell Culture Technologies. And this is going to be the last problem solving session for this course. And there will be another session that will be taken by other PMRF scholars in the coming Tuesday. But from my side, this is going to be the last session. So let's move on to the first question. So the first question is, during depolarization, membrane potential does it shift from negative to positive or positive to negative, or it remains unchanged, or none of the above? So if anyone knows the answer, they can type it in the chat box. Then I'll discuss in detail about membrane potential depolarization and what is the charge distribution. So for this question, the correct answer is that during depolarization, the membrane potential shifts from negative to positive. So there are some important terms which like we will need to know before I go on towards explaining what exactly is membrane potential and how does this change in potential take place. So basically, the term that we encountered in that question was depolarization. So what does depolarization mean? So basically, depolarization means when your positively charged sodium ions, they come inside by using this sodium ion voltage gated channel. So this depolarization is actually a rising phase. Rising, why is it called a rising phase? That is because your membrane potential shifts from negative to positive. And then there is another phenomena which takes place after depolarization, that is repolarization. So as the name suggests, repolarization. Repolarization will be opposite of depolarization. That is, your membrane potential is going to shift from a positive to a negative potential. So since it is opposite of depolarization, it is also known as the falling phase. And what will happen in repolarization is that in your depolarization, the sodium voltage gated channels are opening, like these channels are opening, but In repolarization, these channels will be closed. So the voltage channels, these will be closed. And when these sodium voltage gated channels are going to be closed, then what will open? Your potassium gated channels will open. These will open. Okay. And then what is action potential? So action potential is basically a brief reversal of your membrane potential. So here, during depolarization, what will happen is your membrane potential is going to change from a resting potential of somewhat around minus 70 millivoltage to plus 30. And that is why we are saying that this potential is changing from negative to positive. So now we can discuss like in detail like what is the membrane potential, what is the falling phase, what is the uh, rising phase, depolarization, repolarization, etc. So now here, if you look at this first graph, just one second. Yes. So if you look at this first graph, here you can see that on this axis, there is this distribution from minus 70 millivoltage to plus 40, plus 30, plus 40. Okay. Now, this is going to be your resting stage. This from here to here, which you are seeing. And here on the x-axis, we have time in milliseconds. So this is going to be your resting phase. Resting phase is when there is no stimulus from the outside. But now suppose your neurons have gotten a stimulus, which is here at this point. Now, once they have received the stimulus, then within a span of just a few milliseconds, what will happen? Like your membrane potential will start to rise. And so this is the rising phase or the depolarization phase, as you can see here. So here in the first stage, you are receiving a stimulus, like the neurons are receiving a stimulus, and then as a result of that stimulus, this depolarization event or this rising phase is taking place. Why rising phase? Because the potential is rising from minus 70 MV to somewhere around plus 30 to plus 40 MV. And depolarization because the polarization of the membrane, that is whatever was the polarization state of the membrane, that is broken. It's depolarized. It's not the same as what it was before. It is something new now. It is different now. That is what depolarized. So that is why it's rising now. So this is the rising phase, which you can see in both the graphs, and then it reaches a peak. As you can see here, this peak, it reaches a peak. That peak is achieved somewhere between plus 30 to plus 40 MV. Once this peak is achieved, then what happens? The potential starts to fall, or what is something which is called repolarization. Okay, so now this potential will start to fall, that is repolarization, and again it will go to that uh, resting stage. So now, why this is this depolarization, repolarization, and why are all these taking place? So what happens during depolarization is that, as you can see here, it's written sodium ions in. So your sodium ions are coming inside through the membrane. So what happens in depolarization is that these sodium sodium ions suddenly rush inside using the voltage gated sodium channels which are present in the neuron. And so, like, as soon as your sodium ions are going to rush inside, what will happen? The membrane is going to reverse its polarity, right? And after that, like, once the membrane is going to reverse its polarity from a negative polarization, it's going to become a positively polarized membrane. But once it has reached a certain standard, a certain value, a certain height, then after that, what is going to happen? It's going to fall. And that falling is going to take place because of the closing of sodium channels. And like, so if the sodium channels are going to close, then what will happen? Like, as soon as your sodium channels are going to close, what will happen? Your potassium channels are going to open. And because the sodium channels are closed, so there is no influx of positively charged ions, and the potassium channels are opening. So now what will happen? The potassium ions which are present in your membrane, they will rush out of the membrane. So now whatever the positive charge is present in the membrane, it's going to seep out. It's going to go out through closing of sodium channels and opening of potassium channels. And then what will happen? So again, this localized negative membrane potential of the cell membrane is going to be restored, and it's again going to fall down to the same threshold level, to the same level. Okay. Now there is another state which is called hyperpolarization. So what is hyperpolarization? So what will happen in hyperpolarization is some of your potassium channels will remain open. Like once it has reached this resting stage, the potassium ion channels should close, right? But it sometimes happen. It's a biological system. It's not going to be entirely perfect, right? So what will happen? Some of your potassium ion channels will remain open, and by the time your sodium ion channels are resetting. So this period of increased potassium permeability, because it's still open. So what will happen? If it is still open, then more and more of your potassium ions are going to go out of your cell membrane. And even when the membrane potential was supposed to stop at minus 70 millivolt, now what will happen? It will go on falling down more and more. Okay, because the potassium ion channels are still not closed. So that state is called hyperpolarization state. And once these close, then again it will reach the resting stage where it is supposed to be. All right. So this is it. Yeah. Next. So next, since the question is on depolarization, I'll discuss depolarization in greater detail. So basically, as the name suggests, depolarization, as I mentioned earlier. So this will be the reversal of your polarity, right? Whatever the membrane potential was before, it's going to change, and it's going to be removed. That is why D. Okay. So now, what will happen is, so this repolarization, all this propagation of your action potential is going to take place at the membrane of your axon. Okay. So what will happen? So this is your extracellular environment. This is your intracellular environment. Okay, ECF is these are your extracellular factors, and these are your intracellular factors. And this is your membrane. Now, as you can see, these here, these are your ion channels. So this is your sodium ion channels. And when the sodium ion channels are going to open as a result of depolarization, because there was a stimuli which resulted in the opening of these ion channels, then what will happen? There from outside there will be an influx of the sodium ions inside. So once there will be influx of sodium ions inside, as you can see, so previously there was negative charge all around here, but as more and more sodium ions will come inside, what will happen? Like these negative charge will be neutralized, but as they will come, keep coming inside, it will become a positively charged uh, situation, right? And then once abundant amount of sodium ions have come inside, such that the membrane potential is somewhere around plus 30 millivolt to plus 40 millivolt. After that, what will happen? So these are the potassium channels. What will happen? These sodium ions will start to close. As you can see, these have started to close, and your potassium ion channels will open. So when potassium ion channels will open, what will happen is your potassium ions will go outside the cell. From inside, they will go outside, and that is how your membrane potential is going to be restored. So now here in this graph, you can actually appreciate what like what is the time frame in which your sodium channels or your potassium channels are opening and closing. So here, as you can see in this resting stage, again this is the minus 70 millivolt is the resting stage, and now within the span of just this much millisecond, you can see the scale bar here. Within the span of just this much, like this is approximately around just 1 millisecond, your sodium ion channels are going to open. So your sodium ion channels are opening, and then as soon as this height is reached, then what will happen? Your sodium ion channels will close, and simultaneously your potassium ion channels will open. Now, once this particular dip has taken place, once this hyperpolarization is also done, your potassium ion channels will start to close, and again the same membrane potential will be restored. So now here you can appreciate that within just a time frame of just 2 to 3 milliseconds, all these biological phenomena are taking place, and whatever depolarization, repolarization, restoration of the membrane potential, all that is taking place within just a time span of 2 to 3 milliseconds. So here you can see how, like, what is the direction of travel of the action potential. So this is your neuron. As you can see here, this is your neuron. This is the axon. This is the axon terminal. So your action potential is going to travel in this direction, in the direction of the arrow. So now in the first case, what will happen? So once your soma has received the signal through these dendrites, what will happen? The axon will become depolarized. So as you can see here, suppose just in this part of the axon, like from negative, the like charge has become positive because it is depolarized, but this entire part still has not become depolarized. So now your signal has to travel, right? Like, so signal is received here, like from here it reaches the soma. From here, like the signal has to travel. So the signal travels like this to the axon terminal. It doesn't travel like this, not in just one step. All right, it travels in these small jumps. So as you can see here in this diagram, just one part has become depolarized, and this part is still in its resting potential stage. Now slowly what will happen, this depolarization state is going to spread along the entire length of the axon in small jumps. So but once this depolarization has to travel to another stage, the part which was depolarized is going to become repolarized. That is, the membrane potential is going to like again restore. So here at first the sodium channels of this part were open. Now these will close and your potential will travel to another part where another sodium channels are opening, and again the rest of the part is in resting phase. So this is how in small jumps it is going, this action potential is going to travel, the signal is going to travel. It's not that it just like the action potential doesn't travel from the soma directly to the axon terminal in just one jump. It travels through multiple jumps. So that brings us to our second question, which is, during repolarization, membrane potential shifts from, so does it shift from negative to positive or positive to negative, or does it remain unchanged, or none of the above? So I hope, since I have already explained in the previous question, now you can answer this question. If you can, please write your answer in the chat box. Yes, so those who have answered, it is correct. It's B. During repolarization, the membrane potential is going to shift from positive to negative, and depolarization will be negative to positive. So again, this is just a schematic to make it clear what repolarization is. So what will happen? So this is your extracellular space, and this is your intracellular space. So during repolarization, as you can see here, your sodium channel is going to be closed. As you can see here. Hence, there will be no more influx of the sodium ions. However, your potassium ion channels are going to open, so there will be an outflux of these potassium ions, and that is how the membrane is going to restore its potential. So again, this is just a schematic to show, so that you can appreciate what is the charge distribution. So during the resting phase, the resting potential, the membrane charge is going to be negative. During depolarization, it's going to be positive. During repolarization, it's going to invert and become negative, and then the membrane is again going to come back to its resting potential stage, which is the negative charge. So again, we have this question: during depolarization, which ions move inside the cell from outside? So do sodium ions move inside? Do potassium, or do both of the kind of ions move inside, or none of the above? So if you know the answer, you can write the answer in the chat box. Yes, so the answer is A. During depolarization, your sodium ions are going to move inside the cell from outside. So I have another question. So this is kind of a detailed question. So here you have different stages of the action potential, and each stage is assigned a number, as you can see, 1, 2, 3, 4. Now you have to read these stages, and then you have to assign correct sequence to these stages. Suppose if you say that 4 is going to come first, like that. So we can take 5-6 minutes to solve this question. I'll just read out what are the statements. So the potential difference across the membrane reaches plus 40 millivolt, and the voltage gated sodium ion channels close. So this is not going to be the first stage, right? Because this is what? This is something where that, like, I hope you remember the graph I just discussed. So here it's going to reach here at 40 milliwatt, but this is not the first stage, right? So the next stage which is mentioned is the energy of a stimulus triggers some voltage gated sodium ion channels to open, and sodium ion channels diffuse into the axon cytoplasm. So now here you're getting the stimulus. So I'll just number here according to what is like the stage number which is written here. So this point here is the first statement. Then the second statement is the energy of a stimulus is going to trigger to open some voltage gated sodium ion channels, and then sodium ion channels are going to diffuse into the axon cytoplasm. So your stimulus is going to be here, and after that your depolarization is going to take place. Okay. Then the third statement, more potassium ions diffuse out of the axon until the membrane is hyperpolarized. The potassium channels close. So this hyperpolarization is the step which is here. So this is the third statement. The second will be somewhere around here. The fourth statement, the axon membrane has a resting potential of minus 65 millivolt. Voltage gated sodium ion channels are closed. So now this axon membrane has a resting potential of minus 65. So now this can be either this stage, fourth, or this stage. Okay. So next statement is the voltage gated potassium ion channels open and potassium ions diffuse out of the axon. Alright. So again this will be the repolarization state. So this will be the fifth state. And then more voltage gated sodium ion channels open and more sodium ions diffuse into the axon. So this will be the depolarization state. So this will be here. So now the answer can be like if we are going to take this point from stimulus to be the first like stage, then your first stage will be the second stage where your energy of stimulus is going to trigger the opening of voltage gated sodium ion channels. All right. And now if you look at the graph here, then once your sodium ion channels are open, depolarization will be the next stage. So the sixth statement is going to be the next statement. Then what will happen? Like once it reaches a height
Video
of 40
cut பண்ணு முடி
millivolt or 30 millivolt, then that will be the first statement. Then again your repolarization is going to take place, which is the fifth statement. All right. And then what will happen? We will have this hyperpolarization, which was defined by the third statement. And then your axon membrane is going to get a resting potential. So it is the fourth statement. So this will be the sequence of event. If someone has solved it that the fourth statement is first, then that answer is also correct. So both these answers are correct. So let's move to the next question. Neuromuscular junction is made at junction of motor and muscle and motor neuron, glia and motor neuron, hippocampal neurons and osteoblasts, or none of the above. So I discussed about neuromuscular junctions in I think the week 6 lecture. So who all have answered option A is correct. So neuromuscular junction is basically uh, made at the junction of muscle and motor neuron. So it's clear from the name itself, neuromuscular, right? So it will be a junction between neuron and a muscle. So this is how a neuromuscular junction looks like. So so basically, what is a neuromuscular junction? It is a specialized kind of synapse. All right. So it is a special kind of synapse, and it is not a synapse between two neurons, but it's kind of a junction between a neuron and a cell. So basically, these neuromuscular junctions or these NMJs, what is their role? So they are used for communication between the motor neurons and the muscle cells, which are used for contraction and movement of your muscles.
हाँ.
All right. So here again we have like different action potentials which travel from the neuron to the muscle and again from the muscle to the neurons. Okay. So like these are the ways in which signals travel in our body, right? From neuron to neuron and from neuron to muscle and muscle to neuron. So the next question is, bungarotoxin causes paralysis by blocking potassium channels, acetylcholine receptors, both A and B, or none of the above. Yes, so people who have answered B is correct. This bungarotoxin is a snake toxin and it causes paralysis by blocking the acetylcholine receptor. So I'll discuss in detail about this toxin and like how does it come. So this is a neurotoxin because it blocks the acetylcholine receptor. So that means it affects the neurons. All right, and it is a very potent toxin because as you can see here, it's 16 times more potent than the cobra venom, and this its LD50, that is its lethality, is 3.5 microgram, which is considered to be something which is very high. So it's very powerful and it's going to induce muscle paralysis because it's going to block the acetylcholine receptors. And if you remember from the last lecture, the acetylcholine receptor clustering takes place at the neuromuscular junction. Okay, so since it acts on those, it induces muscle paralysis. Okay, and the venom that is secreted by this bee candida snake does not consist of just the bungarotoxin, but it also consists of many other kinds of neurotoxins. And then what does this venom do? It affects the ability of the neuron endings to properly release the chemical, and that chemical is again acetylcholine that sends the message to the next neuron. So basically, it blocks. Sorry, is someone not on mute? So I request everyone to please be on mute. I have muted that person who was not on mute. Thank you. So basically, what happens is it's going to not allow the neuron endings to release acetylcholine. Okay. So what will so neurotoxins, as the name suggests, they are going to target nerve cells or neurons, and they mostly target the motor neurons which are in the peripheral nervous system. So as you can see here in this diagram, the peripheral nervous system or the PNS is extending through the entire body. So if there is going to be a toxin or there's going to be a molecule which is going to block the motor neurons, then the entire like your peripheral nervous system is going to collapse. So that is how it causes paralysis. Okay. So now, as I mentioned earlier, these neurotoxins specifically target the neuromuscular junction. Okay, and because they are targeting the neuromuscular junction, they are going to block the communication which is happening between your neurons and your muscles. So if that communication is not going to take place, you are going to get paralyzed, right? Because then your muscles will not know how to move or how to react in that particular situation. Okay. So now uh, we discussed this earlier that fake neurotoxins target the motor neurons, and so there are two different kinds of neurotoxin: beta neurotoxin and alpha neurotoxin. So these beta neurotoxins, what do they do? They act presynaptically, all right, and these alpha neurotoxins act postsynaptically. So your beta neurotoxins are going to prevent acetylcholine from being released because they are acting presynaptically, like before the. So this gap is the synapse or synapse, right? So they are going to act presynaptically. That means just before the synapse, they are going to block these acetylcholine receptors, and your alpha neurotoxins act postsynaptically, that is after the synapse. So again, this brings us to like the neuromuscular junction. So this is the presynaptic terminal that I was talking about. So your beta neurotoxins are going to act on these presynaptic junctions, and your alpha neurotoxin are going to travel through the synapse and act as this postsynaptic junction. Okay, so this is the acetylcholine receptor which has been clustered here, and your bungarotoxin is one alpha neurotoxin because it blocks these acetylcholine receptors which are present on your like NMJ, the neuromuscular junction. But your bungarotoxin is going to travel through the synapse so that it reaches the acetylcholine receptor. So this is the structure of the bungarotoxin. As you can see, it's a very it's not a very big protein. It's a small peptide actually. And as I mentioned in the diagram previously, this bungarotoxin acts postsynaptically. So it is an alpha neurotoxin. So there are two different kinds of bungarotoxins. It can be either alpha bungarotoxin or beta bungarotoxin. So your alpha is going to act postsynaptically, and your beta is going to act presynaptically. So when it's acting postsynaptically, what will it do? It will bind irreversibly to the acetylcholine receptor. That is the acetylcholine receptor which is present. This bungarotoxin is going to bind to it irreversibly. That means it's not going to come off the acetylcholine receptor. Okay. So that is why if this acetylcholine receptor is blocked, it won't be able to transmit signal, and that is going to result in paralysis. And what will beta uh, bungarotoxin do? It will act presynaptically. So if it is going to act presynaptically, then what will it do? It will alter the release of acetylcholine in the presynaptic terminal, and this beta bungarotoxin is not going to bind directly to the acetylcholine receptor, but it's going to stop the release of acetylcholine. And if acetylcholine is not being released, then what will happen? It will not be there to bind to the acetylcholine receptor and transmit the signal further. Okay, so these are the mode of actions of these two different classes of bungarotoxins. So as you can see here, this is the acetylcholine receptor, and here this is the acetylcholine binding pocket. And here in the acetylcholine binding pocket, what alpha bungarotoxin is going to do? It's going to come and bind at this pocket. Okay, and since it is binding irreversibly, even if there are a lot of acetylcholine molecules which are present outside, they will not be able to bind to the acetylcholine receptor. And but if we talk about the other, again this alpha bungarotoxin is binding to the acetylcholine receptor which is present at the NMJ. So now moving to the seventh question. So for myotubes cultured on cantilever devices to move cantilevers, which property of cantilevers should be similar to myotubes? So is it stiffness, color, weight, or width? So here the correct answer will be stiffness. I will explain now. Basically, what are cantilevers? Cantilevers are like they are something which are supported on just one end. So here this will be your cantilever. Since this is a beam, so this will be a cantilever beam. So cantilever is nicely supported on one end and it's free on other end. Okay. So here in this schematic, so basically here you can appreciate how a cantilever is used to measure muscle force or muscle stiffness. So this kind of cantilever device is actually an MEM. So I think in second or third class we discussed about MEM. If you don't remember, I'll suggest you go and revise it once. So basically, this cantilever, myotubal cantilever device is kind of an MEM, or more specifically, a bio-MEM. Okay, so here we have our cantilever. This is the cantilever, and you can see it's just supported at this end, and it's free at this end. This end is free, and we have our myotube here on the cantilever. Okay, so now then we have the patch clamp electrode like here, and this myotube is in this stimulation chamber. So now whatever stimulus we are going to provide to the myotube, it's going to move, right? And how is it going to move? It's going to move because of its stiffness. Depending on its stiffness and depending on the kind of stimulus provided, it's going to act. Okay, and uh, then accordingly, just as this myotube will move, whether it will contract or otherwise, this cantilever is also going to like move along with it. And then when this cantilever is going to move, this laser deflection, so the laser will be deflected, right? It will not be at the same place where it was before if the cantilever has moved because of the as a result of the contraction of the myotube. So this laser deflection will happen, and this laser deflection can then be detected by this photo detector, and this information then can go to your computer or to any of your processing software, and then you can determine like whether your myotube has contracted or not as a response to the stimulus that you were providing. Okay, so here now you can see, you can appreciate it more. So this is the cantilever. Okay, so this is the cantilever. You can see it's just supported at one end and not on the other. And there, your myotubes are going. Okay, so now if there is a passive force, there is a small passive force, then the displacement of this cantilever is only y1. However, when there is a contractile force, which you can see here, then this displacement is y2. And so as you can see here, the cantilever is displaced by y1 and y2 distance. So this displacement of the cantilever can be detected by the deflection of the laser, which you can see here. This laser is coming here, and then we can decide, like we can conclude whether a particular kind of stimulus is resulting in the contraction or the relaxation of the myotube. So this is how, using a cantilever, we can determine what is the force that is being experienced, or that is by a myotube. So next question is, hippocampus is associated with movement, color vision, learning and memory, or vocation. So we discussed about hippocampus and different regions of the brain in the previous lectures, right? Yeah, so option C is correct. Hippocampus is associated with learning and memory. So next question. So this compound is a pungent pain producing compound found in plants of the capsicum family, and this compound exerts excitatory, desensitizing and toxic effects on a subset of sensory neurons, including the polymodal nociceptor population. So which is this compound? Yeah, so most of the people have answered correctly. It's this option A. It's like kind of evident from the question. Capsicum family will plants of capsicum family will produce a compound which is like capsaicin. So this is the capsaicin molecule, and there was another thing in the question, these polymodal nociceptor. So nociceptors are basically the pain receptors. Okay, so this is the like chemical formula of the capsaicin compound. So now here you can just see, like this is just for your information, what happens when you eat a pepper or a chili or capsicum. So this compound capsaicin which is present, it's going to bind to the pain receptors on the tongue. So pain receptors or nociceptors, and then what will happen? So when it's going to bind to the pain receptors, you are going to experience like that it's hot, it's chilly. Then what will happen? Your brain will try to cool the body by, like, in response to these pain receptors, the brain will try to cool the body by sweating, salivating, etc. And then again, when the capsaicin is going to reach the esophagus, then again it's going to cause a burning sensation in the chest or the lungs, and then then the subsequent things will happen. Okay, so it is because of the binding of this compound capsaicin that we have the different responses that we get when we eat pepper or chili. So these are the nociceptors, like basically these are the kinds of nociceptors. So basically your heat receptors, which is the TRPV, or the cold receptors, and then the polymodal receptors, which can either be peptidergic or non-peptidergic. So as the name suggests, peptidergic means it's going to bind to some small peptides, and non-peptidergic means it doesn't need small peptides to get stimulated. So this brings us to the last question. Which of the following, which of the following is a calcium ion channel blocker? So is it TTX, 4AP, calcium, or free radicals? So which of these block the sodium channel? Yes, so here the correct answer is A, TTX. So this TTX is actually something which is called tetrodotoxin. So basically what it does is that, so it mimics a hydrated sodium ion. So this is a hydrated sodium ion. So this sodium channel will open when it interacts with this sodium ion, right? So this TTX is going to mimic the sodium ion, and then it is going to block the ion channel. So as you can see, it's like fairly big in size. So because of that, what it's going to do, your sodium ion will not be able to, like your small sodium ions will not be able to compete with this bulky TTX, and that is how like it blocks the sodium channel. So that is all for today's session. So this is the last session for this course, like for this uh, like this duration. I am sharing the link of the YouTube channel, and all the lectures have been uploaded there. So if you want to revise or if you want to get your doubts cleared, you can like go to that YouTube channel. And as for the exam pattern and everything, I'm not sure because like we are here just to deal with these problem solving sessions. We are already given these set of questions so that like we can solve those for you. These sessions are just supposed to help you analyze which kind of questions you're going to get, or like how you should solve questions. So it's just for that. As for exam pattern, I'm not sure. You can like mail the NPTEL help site, and from there you can get. So thank you all for attending the session.